So what can we use ATP for? Why is it useful to us? So the main function of ATP is to release energy by breaking the phosphoanhydride bonds. So we can use this energy from the ATP to drive chemical reactions that are required to be performed within cells. Another use for it is the phosphate that is actually released when we hydrolyze ATP to ADP. So this phosphate can actually be added on to another component. For example, we see here that glucose can be converted to glucose 6-phosphate by using the phosphate that has been removed off ATP. So we can have other bases forming these nucleotide triphosphates, so it's not just adenine. So we will see our UTP in a later lecture, and this is involved in a process where we're actually building a storage of glucose called glycogen. So the UDP is actually used in a process called glycogenesis. So we'll also see that GTP is created in another one of our metabolic reactions. This is in the citric acid cycle, and the CTP is also involved with the metabolism of lipids. So we're going to talk about the first of our coenzymes now. So remember, the coenzymes are really important to a lot of metabolic reactions, and we need the coenzyme to be added to our apoenzyme, and this gives us our functional enzyme, which can drive the reaction forward to create the products. And our functional enzyme has the name of either a conjugate enzyme or a holoenzyme. So this is where we have the coenzyme and the apoenzyme combined. So the apoenzyme part is the protein-only part, whereas the coenzyme contains other groups that are needed for the reaction. So let's have a look at what some of those groups are for our flavin adenine dinucleotide, or FAD. So we see here that the FAD is a coenzyme that's required in many of our metabolic reactions, and it's actually from riboflavin, which is vitamin B2, that we're able to derive our FAD. So the riboflavin group is actually modified into flavin, which is a heterocyclic amine, and this is the component that is needed for our FAD. So we also attach the flavin to another group called ribitol, which is an alcohol derivative of our ribose. So we've added more alcohol groups to our ribose sugar. So we'll see this structure on the next slide. And then the last part of our FAD is our ADP. So we can see the groups that are making up ADP here in purple. So we've got the adenine, the ribose, and the 2-phosphates. So just like what we saw on the previous slides where we were comparing ATP, ADP, and AMP, we've got the core structure here of ADP, but the only difference is instead of just having phosphates attached to our ribose, we now go further and start attaching this ribitol and the flavin. So when we have ADP, ribitol, and flavin all connected, that's what gives us our FAD. So here's the structure of our FAD. So we don't need to know it in this much detail, but just to show you exactly what these components are. So we've got our components that we've seen many times before. We've got our purine nitrogen containing base at the end here, being adenine. We're then linking that to our ribose sugar, and linked to that ribose sugar are our 2-phosphate groups. So all of these structures together, which we see down here, is our adenine diphosphate. So we've got our two phosphates connected to the sugar and the nitrogen containing base. So this is our ADP. So we've actually connected the phosphate to another sugar group. So here's our second sugar, which in this case is ribitol, which remember is the alcohol derivative of the ribose. So it's also in the straight chain form rather than being cyclical. And then the last component of our FAD is the flavin, which contains several amine groups. So each of these nitrogens is an amine, and then we've got an amide formed in two locations here as well. So these ones are the amides, and up the top here we've got the amines. Alright, so this is more detailed than we need, but it's worth seeing the overall structure and what it actually looks like. The main things we need to remember about FAD is that it's a coenzyme and that it is derived from riboflavin, and the fact that we have the flavin as a part of the structure. It's also worth knowing the exact structure that's in this pink box. So we've got the ADP, which is linked to our ribitol, and then the flavin. And here we can see the entire structure drawn out. So how does FAD actually function? So it actually drives what we call redox reactions. So remember, in a redox reaction, we have one component being oxidized and one component being reduced. So the oxidation and reduction reactions that are going to be driven by FAD, we're going to be looking for the loss and gain of hydrogen. So if something's being oxidized, then we're losing hydrogen. And also there will be electrons lost because the hydrogen actually contains electrons. Whereas if something's being reduced, then it is gaining hydrogen. And if it's gaining hydrogen, then that means we're actually gaining electrons as well, because that hydrogen has an electron associated with it. So the really nifty part of our FAD is that it can actually be in two different forms. So it can either be in the oxidized form or the reduced form. So this actually allows it to participate in reactions where it's going to be oxidized or it's going to be reduced. So it can do both types of reactions. So if it's in the oxidized form, it will actually be reduced. So if it's in the FAD form, it will gain hydrogen. And by gaining hydrogen, it's going to take the hydrogen off another component. So this will be a component within our metabolic reaction that is going to give hydrogen to the FAD. So that's why we say that the FAD is an oxidizing agent, because it's actually going to oxidize another component by taking its hydrogens. And through this reaction, we change FAD into FADH2. So that's what we can see summarized down the bottom here. We have the FAD, and it's gaining 2 H plus and 2 electrons. So this is going to be from another biological molecule involved in some sort of metabolic process. So we're taking these hydrogen atoms off the biological molecule and putting them onto our coenzyme to create our FADH2. So when the FADH2 is formed, this is actually now the reduced form. We call it the reduced form because it is the hydrogen-rich form. So we can think of the FADH2 as being hydrogen rich. It has lots of hydrogen compared to the FAD, and also electron rich. Compare this to the FAD, which is actually electron poor by comparison to FADH2, and also hydrogen poor. So we can actually interchange between these electron rich and poor versions through undergoing these redox reactions with other biological molecules which are used in metabolism reactions. So what can the FADH2 do? It can actually be oxidized. So it can go from FADH2 to FAD. So we go in the opposite direction, the reverse reaction here. And in doing this, we're actually going to be donating these two hydrogen atoms onto another biological molecule. So this is what we call the act as a reducing agent. It's causing another biological molecule to be reduced. So if we have our FADH2, then this is going to give hydrogen plus electrons to a biological molecule. Whereas if we have FAD, it's the opposite. It's going to take hydrogen and electrons from a biological molecule. So we'll see what these biological molecules are once we start actually looking at our metabolic reactions. So the really unique thing about these coenzymes is that they can continuously change between these oxidized and reduced forms. So it can interchange between the FAD and the FADH2. And the other thing that this allows it to do is when it's in the electron-rich form, which is our FADH2, it can actually function as an electron carrier and carry these electrons to the electron transport chain where they are used to drive the production of ATP. So here we can see exactly what happens in this reaction in terms of what modifications there are to the FAD structure. So we've got our FAD on the right here, and that form is actually going to accept two hydrogen atoms. So now instead of having these double bonds, which we see on the left-hand side here, we've actually broken those double bonds, and instead the nitrogen bonds to an extra hydrogen. So that's what gives the FAD the capacity to take on these two hydrogens. There's these extra double bonds on the left-hand side which can be broken to instead form bonds with the hydrogens. So the FAD can happily exist in either of these two forms. If it's the FAD, then we call this the oxidized form because it is hydrogen and electron poor, whereas FADH2, which is the reduced form, is electron and hydrogen rich. So we can see what we can actually do with our FAD down the bottom here. For example, if we have a compound that is saturated, so this tells us that it has the maximum number of hydrogens, we can actually take some of these hydrogens off our biological molecule and give them to FAD. So this is a redox reaction. In this case, the saturated compound is being reduced and is being oxidized, and FAD is being reduced. So we reduce FAD and we oxidize our saturated compound. So although they're in the oxidized and reduced forms, it actually does the opposite in the redox reaction. So the FAD has the capacity to take on an extra two hydrogens, and those hydrogens come off our saturated biological molecule. So by doing this, we actually are creating an unsaturated biological molecule that now needs to form a double bond between the carbons here in the absence of these hydrogens that have now been given onto our FADH2. So we can see that by converting FAD to FADH2 that we were actually able to oxidize a biological molecule. So here the biological molecule contained more hydrogen, and then we've removed hydrogen by giving it away to the FAD coenzyme. So the form of the coenzyme will always be different on each side of the reaction. So on one side it will be in the electron-rich or poor form, and the other side it will be the opposite. So this is what drives the reaction, the ability for the FAD to actually either accept hydrogens or donate hydrogens. And it does this by oxidizing or reducing another biological molecule. So NAD functions in very much the same way. So we'll see that NAD actually performs almost an identical function to what the FAD did in terms of being able to strip hydrogens off a biological molecule or donate them by either being the oxidizing part of a redox reaction or the part that's reduced. So again, we've got our ADP, which is the adenine, ribose, and the two phosphates, and then we've got our extra groups added on to the end here, which is what makes it a NAD, or nicotinamide adenine dinucleotide. So we derive our nicotinamide from our niacin, which is vitamin B3. So we modify this slightly to form our nicotinamide, and by adding our nicotinamide and our ribose onto the ADP, we've created our NAD. So here we can see the structure of NAD. So up the top, down the bottom here, we've got the adenine, the ribose, and the phosphate. So together, this part of the structure gives us the ADP. So just like with the FAD, it has this ADP core that we then add on to via this phosphate. So in this case, the extra sugar is a ribose. Remember, for the FAD, it was the ribitol, which contained more alcohol groups and was not cyclic, it was actually a straight chain. And the last part of our NAD is the component that's derived from our vitamin B3, niacin, and that's our nicotinamide. So we can see the nicotinamide contains two different functional groups. We've got the amide functional group up the top here, and then we've also got an amine in the ring structure here. So just like our FAD, the NAD is able to exist in these two different versions. So that's why it's actually able to participate in these redox reactions just like the FAD could. So the NAD plus is what we call the oxidized version because it lacks hydrogen. So we can say that this is electron and hydrogen poor. So this is the component of the reaction that will act as the oxidizing agent. It's going to accept H+. So the other version that we can have is our NADH, which is what we form after the NAD plus has accepted hydrogen. So we call this the reduced form, which is electron and hydrogen rich. So we've got more electrons and hydrogens than the NAD plus. And this one can actually lose the hydrogen and electrons to go back to the NAD plus form. So we can see the ways that these reactions can occur down the bottom here in the summary. So if we are going in the forward direction, we're actually combining NAD plus with hydrogen atoms, and this is allowing us to form the reduced version, which is the NADH. We'll also release an H+, so we can actually only take on one additional hydrogen instead from what we saw in the previous one with FAD that could take on two additional hydrogens. So when we're going from left to right, we're going from the oxidized form of NAD to the reduced form. So that means that the biological molecule that we're taking these hydrogens off is actually being oxidized by our NAD+, which is itself being reduced because it's gaining hydrogens. And we can go back in the other direction as well, where we're doing a oxidation reaction to go from the electron and hydrogen rich form, NADH, to the NAD+ form, which is electron and hydrogen poor. So just like our FAD, the NAD will continuously change between these oxidized and reduced forms when it participates in these redox reactions. So we'll see that the NAD is extensively used in a lot of metabolic reactions, especially in the citric acid cycle. And it also has the function of carrying electrons to the electron transport chain. So which of our forms of our coenzyme here do you think would be the carrier of the electrons to the electron transport chain? Would it be NAD+ or NADH? It's actually NADH, because this is the electron and hydrogen rich version. So we can take these electrons and offload them into the electron transport chain.